Hello, everyone. I'm Susanna Whitacus-Weaver. I'm the director of the Wisconsin Center for Origins Research, and we welcome you to our fall WiCOR lecture. So if you want to learn more about WiCOR and our research trying to understand the origins of life on Earth and how life might be out there elsewhere in the universe, we have the QR code up here for you to go take a look at our website, follow us on social media, and we have a pamphlet and some other materials outside if you'd like to take that information home. So with that, I'm going to turn over to my colleague John Yin, who will introduce tonight's speaker.
Thank you, Susanna. So I'm John Yin. I'm a professor of chemical and biological engineering here at the UW, and also one of the founding faculty of the Wisconsin Institute for Discovery. I'm delighted to welcome you tonight to what promises to be a memorable event. And before I begin, I should thank our sponsors, which include WiCOR and the Wisconsin Institute for Discovery. So it's an honor to introduce our speaker, Dr. Jim Cleaves, professor and chair of chemistry at Howard University in Washington, D.C. Dr. Cleaves is a leading figure in the study of life's origins with research spanning astrobiology, complex reaction networks, and the chemistry that may have set the stage for life on our planet and in other worlds. Dr. Cleaves earned his PhD at UC San Diego under Stanley Miller, a pioneer in prebiotic chemistry. He's held research appointments at the Carnegie Institution, the Institute for Advanced Study in Princeton, and the Earth Life Science Institute in Tokyo. His work blends laboratory experiments, theory, and broad interdisciplinary collaboration to explore how simple molecules become the building blocks of life. So Dr. Cleaves has also shaped the field through leadership and public engagement. He served 3 terms as president of the International Society for the Study of the Origins of Life, and he's organized major conferences around the world, published widely in both scientific journals and popular formats, and he's deeply committed to mentoring and education at Howard University. Please join me in welcoming Jim Cleaves with a Wisconsin welcome.
Thank you so much for the very kind introduction, and thanks to the WiCOR staff and leadership for inviting me up here. I appreciate it. I was trying to remember on the way coming in here when the last time I've been to Wisconsin was, and it turned out it was 1987. I came up, I took a Trailways bus from Philadelphia to see the Grateful Dead, right? So I had a good time in Wisconsin. All right. Oh, okay. One second. There we go. Okay. So what I try to talk to you about today is kind of a broad overview about how kind of thinking about the origin of life has changed over time. We wrote a book about the history of this idea. It came out maybe about 7 or 8 years ago, called A Brief History of Creation. And it was kind of, it's really interesting how the conception of what life is and how it might come to exist has changed over the last few centuries or even millennia, right? We have kind of metaphysical, supernatural explanations in a lot of religions. But it's not until really sometime in the early to mid-Renaissance that people start to really drill down on this in a experimental fashion, right? A carefully controlled fashion. So we'll get some of that, and then we'll look at some of where we are today with this question. It's a really fun question because it is not only so important to all of us, but it still remains just a really deep mystery in a lot of respects. So this is the big question, right? How do we go from a planet early after its formation that is a violent, hot, desolate place with no life on it to one like the one we live in today that is just absolutely teeming with every kind of biology, right? Almost every inhabitable niche on our planet is filled with living things. It's hard to find a place where there is no life on our planet. Part of that is the domain of evolution, right? Understanding how things change over time and become new species. But when you trace that back, you get back to this fundamental question of what was the first living thing, right? And what was the process that allows that transition? And so I like to start, this is a quote from my advisor's book that came out in 1974 that he co-wrote with Leslie Orgel. They were both big luminaries in the early scientific field. It must be admitted from the beginning that we do not know how life began. It is generally believed that a variety of processes led to the formation of simple organic compounds on the primitive Earth. These compounds combined together to give more and more complex structures until one was formed that could be called living. No one should be satisfied with an explanation as general as this, right? And you know, here we are 51 years later. It's hard to say that we're in a more specific kind of place than this, right? In some respects, yes. In others, the questions have changed and things are changing. But it's really a profoundly difficult problem, and it's been acknowledged as such for some time. Okay, so before we even get into this whole thing, there's this question of what is life? And I like this quote from Supreme Court Justice Potter Stewart. I shall not today attempt further to define the kinds of material I understand to be embraced within that shorthand description, and perhaps I could never succeed in intelligibly doing so, but I know it when I see it. And he was, of course, referring to pornography, not life. But unfortunately, a phenomenon that people really have different opinions about, and it's hard to empirically define. This is what life is. Some people believe viruses are alive, some people don't. There are hybrid organisms that it's hard to say how they are alive or at which boundary levels. So generally speaking, people believe it's something that can make copies of itself and evolve over time, right? Make mistakes that are adaptive and selectable. So going back, deep in at least Western cultural traditions, we have a creation myth that explains where every living thing on Earth came from. It's in the first page of the Bible, right, in Genesis. But it's interesting to think about that. This is, obviously the Bible is that part of the Bible is maybe 3,500 years old, somewhere in that neighborhood. People, of course, were well familiar with the concept of having babies, right? I mean, it's something you could observe. Half the people would literally experience it, right? So that couldn't have been a mystery as to where people came from, right? They came from other people. You get into the era of agriculture and animal husbandry, obviously people understood that you sow a certain kind of seed, you get a certain kind of plant. If you want to raise goats, you put two goats together. But of course, there wasn't the concept of species evolving over time, right? Those could still go back to the original, specially created versions. All right. And we see things like this kind of mentioning the concept of spontaneous generation, so the de novo existence of life, or creation of life in the environment, coming up also in the Bible. The plague of frogs that struck Egypt is believed to be referencing the fact that there's a species of frog that has very, very small eggs. And so people had not well connected the concept of those frogs hatching from those eggs. All right, it seemed to be a spontaneous phenomenon. So, I mean, a lot of this belief, again, is due to the lack of a careful kind of naturalism, right? So some animals, people, goats, sheep, so on, clearly came from previous animals. But some forms of lower animals, insects, worms, it's thought maybe spontaneously arose from decaying matter. And we see this kind of thinking extending all the way up into the early Renaissance. This is Johann von Helmont. And he was kind of an alchemist, right? Nascent chemist in that development of the field, had written a recipe for how he believed mice were created that involved taking dirty laundry, adding some grain, wait 3 weeks, and you are going to have mice, right? So this is, at least it's an experiment you can do. You feel bad for his mom, right? But right about the same time, we start seeing the first really controlled explorations of this concept of spontaneous generation. Like science is becoming codified practice. And Francesco Redi, who was a court physician in Florence, was getting into arguments with people about this concept of spontaneous generation. And it was believed that maggots spontaneously generated in rotting meat. And so he did a simple experiment. He put some meat in a jar and put a lid on it, and there were no maggots, right? And then people said, yeah, but the thing is, there's no air, and this process needs air. So he conducted another experiment in which he sealed the flask with a piece of cheesecloth, right, a gauze, so air could get in and out. But you could clearly see flies buzzing around the top of it and laying their eggs on top of the gauze, right? So this was considered the first scientific refutation of the idea of spontaneous generation. Right? Okay, so now we have an explanation. As Reddy stated, all animals come from eggs or something resembling an egg, right? That there is a continuity of species. Shortly thereafter, we have Van Leeuwenhoek's application of microscopy to the natural world, right? So this is a replica, at least, of the very earliest microscope. The actual thing is only about 5 inches long. It's very small. And what it is, there's a little fisheye lens here. It's about the size of a BB, okay? And then you would impale your specimen on this little spike, and then you could adjust the focal length here and center your sample using this screw. Right? So it's just a little thing, and you put your eye in and from the other side, and you could see things magnified a fair amount. Not a modern microscope by any means, very simple device, but Van Leeuwenhoek started applying this technique to all of these things, and he discovered this microscopic world that no one had even imagined existed, right? He essentially invented microbiology. Right, he would scrape scum off his teeth and put it in there and see things swimming around, pond water, all of this stuff. And at first he thought he didn't want to tell anybody because he thought they'd think he was crazy, right? He was, of course, there's this tiny invisible world nobody else can see but me. But eventually he started writing letters to the Royal Society, and these devices spread and people could confirm it, and this became whole field of study. So now, of course, the question of spontaneous generation got bumped down to the microscopic level, right? That, okay, flies, we see eggs, fine. These things are too small to have eggs. This has to be something else, right? And in between, we had the development of the field of organic chemistry. So Friedrich Wöhler was a German chemist. Early in the development of the field, he found that by mixing two compounds that were understood to be inorganic, and the distinction being that organic compounds only came from living organisms, right? The ones that had been isolated were only known to come from certain plants or barks or things like this. But he found that by mixing two inorganic compounds, he was able to make urea, which is a fairly simple compound. It's technically an organic compound. That until that time was only understood to come from the urine of animals, right? So this was starting to blur the line between needing pre-existing life to make biological products. Right? So this is kind of muddying the water in the other way. It's not disproving spontaneous generation. It's proving that there's a non-biological way to make biological things, right? And so not long after, this is an intense period of exploration around the world, fossil digging, coinciding with the development of the Industrial Revolution, right? People are digging holes to excavate coal. They're finding fossils. They're using the indicator fossils to predict where a coal will be found. without really having a theory of fossilization. But there was a publisher in Scotland named Robert Chambers, who wrote this book that was this sweeping explanation for the development of the cosmos, from the creation of the universe all the way up to the evolution of man. And it was all based on what he had kind of picked through the scientific literature of the time. And he created this beautiful kind of cosmic narrative of creation, right? And how many of you have seen the program Cosmos? Okay. Actually, it's like the 19th century version of Cosmos, right? It explains everything with the to-date science of that time. And this book, partly by virtue of the fact that the British Empire was global, was like the biggest hit book around the world, right? He published it anonymously because he was quite certain that his views in conflicting with the Bible would bring problems for his family. But Prince Albert read it to the Queen every day at tea. Abraham Lincoln got a first edition and read it, and his law partner said he was very influenced by it and warmed up to this idea of basically evolution. This is a diagram from the original version of the book that Chambers made, and what you'll recognize it as is the family tree, right? And so we have here fish, reptiles, birds, mammals, right? And the idea that there's an organism that spins off mutable species that become new branches of life, right? Nobody gives him credit with the theory of evolution because this is kind of a proto version, but there it is, right? It's being laid out. And interestingly, as I said, a very famous British reverend took great offense at this book, right? That it just basically discredited everything in the Bible, and wrote an essay that if this book is true, religion is a lie. Human law is a massive folly founded on nothing than an arbitrary book. Morality is moonshine, and man and women are only better than beasts. But I cannot think but that the work is from a woman's pen. I throw that in on top of it, right? So, but you can see this book, who else read this book? Charles Darwin, right? And you can see that this kind of concept of phylogenetic trees is starting to work its way into the scientific zeitgeist, right? And what's especially interesting, I like this. This is the kind of scrap of paper you find on your desk 4 months later, and you're like, what? But I think this tree, I don't know, but that it has a root, right? And so Darwin was starting to make this, the logical conclusion, there has to be a beginning of this tree, right? The first seed that grows the tree. And it's really interesting to note, of course, On the Origin of the Species was published and republished multiple times. In the original edition, Darwin wrote, as to close the book, there is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that, whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved, right? People gave him a lot of smoke for this because he left out the possibility of the involvement of God in the process, all right? So the second edition, he added a couple words, right? It was, by the Creator. I meant to say by the Creator, right? And then, of course, all the scientist friends were like, you really are a wet noodle, So he took it out in the third edition. So, but here we're starting to see this. It's being recognized as a paradox, right? That evolution makes sense. All life comes from other life. It changes over time, but there has to be a source at some point, right? And although Darwin never really wrote on this publicly, we have correspondence that's been found that was saved, where he wrote a friend about this kind of idea of the prebiotic pond. He said, thinking about this problem, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes. And at the present day, such matter would be instantly devoured or absorbed, right? So this simultaneously explains not only kind of a process for spontaneous generation, but it explains why you don't see it today, all right? That because there is life everywhere now, as soon as this process starts, something else eats the sandwich, right? And that's the end of that. But this is also kind of the first in-print version of this idea of a prebiotic soup, right? That there are chemicals sitting in a puddle that somehow, being acted upon by the environmental processes, complexify and come to life, right? So slightly after Origins is published, there's a very public debate now in France over whether or not spontaneous generation of microbes takes place, all right? Between Félix Pouchet and, most notably, Louis Pasteur. And they go through these same sort of problems of sterilizing solutions and then people complaining that they're not open to the air, therefore this process requires air, and so on and so forth. And they're going back and forth. And what Pasteur finally, ingeniously invented was this special flask design where you could seal a tube, boil it, pasteurize it, right? Sterilize it, and at the end, you could break off the tip so that it's possible for air to exchange with the solution in it. In this case, it's a hay infusion. But any dust containing microbes or anything will get trapped in this little U-neck region, right? And not make its way into the nutrient broth. Fun fact, these flasks have been sitting in a cabinet in the Sorbonne for, over 150 years. They still haven't come to life. They're still sitting there sterile. It's pretty neat, right? And Louis Pasteur was feeling pretty proud of himself. He said, never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment, right? Well, this creates kind of a paradox because we are here. So it seems logical there has to be spontaneous generation somewhere, if it's not on Earth, somewhere else, perhaps in the solar system or beyond. But we don't have anything to go on this, right? Okay. It's worth pointing out at the same time, conceptions of the age of the Earth are changing very rapidly, right? So it's a question of how long does it take for this process to take place? I think what Darwin was pointing out, that you don't see this happening all the time, maybe that it's a long, drawn-out process, or maybe it takes a couple of minutes, but in any event, it's always squelched very quickly. During the 19th and early 20th century, there was a rapid development in the understanding of the age of the Earth, right? And so therefore, the time scales of evolution and the amount of time that was available for spontaneous generation to possibly have taken place. So this is a compendium of estimates for the age of the Earth over the last 400 years or so. And you see a lot of estimates that are based on biblical estimates of generations, times that are reported in the Bible and so on. It's 4,000 years. And then very soon around the time that radioactive decay is discovered, scientists become able to estimate how old rocks actually are based on radioactive dating. And you see very rapidly increases in understanding that, maybe it's a few thousand years, maybe a few 100,000 years, to really deep lengths of time, like billions of years, right? And a lot can happen in billions of years, right? It's an inconceivably long amount of time. The red dot here is the most current estimate. It's about 4.55 billion years old, based on dating of some meteorites that are coeval with the early solar system. Okay, so now these things are starting to come together, right? We have Wohler's concept that you can make organic compounds without biology. You have the idea of a Earth that is old but not infinitely old, right? You have the idea that there was an environment before biology arose that may have been very different than the current environment. And almost simultaneously, two scientists, J.B.S. Haldane, working in the UK, and Alexander Oparin, working in the Soviet Union, both published short works describing their models for the origin of life, which turned out to be very similar. And roughly speaking, goes something like this. The early Earth was lifeless, there was liquid water, the atmosphere was different. The atmosphere didn't contain oxygen because oxygen comes from plants, right? And it was thought at the time the early atmosphere may have been more similar to the gas giants, dominated by hydrogen and methane and ammonia, things like this. And in fact, things like lightning and ultraviolet light acting on that would create this rain of organic compounds that would be falling into the ocean. And those things would self-assemble into cell-like objects and eventually, over time, gain the ability to reproduce themselves, right? And that's how it started, right? They both acknowledge that they came up with the idea independently, and we're fine with crediting each other. Okay, so this is roughly speaking the scheme that Oparin and Haldane laid out. It's called the Oparin-Haldane hypothesis. It's still roughly the model that scientists work with. This idea that you have a period of prebiotic chemistry where the environment is providing compounds that ultimately life can be made from. There is some very mysterious process called the origin of life, where there's an actual transition from non-living to living matter. There's some degree of complexity you could argue about what it actually is. That goes through a period of early evolution to give rise to an organism, a hypothetical organism we call the last universal common ancestor. We'll talk a little bit more about that in a second. And then that early organism radiated out through Darwinian evolution to give you the diversity of the biosphere we have today, right? And so this allows you to ask some kind of concrete questions, right? So things like, well, okay, what was the atmosphere like? And if it was that way, what chemicals would it make? What is the behavior of those chemicals? How do they self-assemble? Things like this. But that's a general schema. All right. So 1953 was a big year for chemistry. We saw the publication of the structure of DNA, which very well explained how Darwinian heredity works. And that same year, let me back up. We'll talk about the same year in a second. Once we understood that, we now had the ability to actually make phylogenies based on organisms that don't fossilize, right? Like microbes, right? And so we're now capable of making full universal trees of life for every organism we encounter and relating them by the similarities of their DNA sequences, right? And so Carl Woese constructed what is the modern accepted tree in the mid-70s, which basically divides the life into three kingdoms: the archaebacteria, the bacteria, and the eukaryotes, right, nucleated cells. And you can see somewhere these three have a common origin, right? So they have a common ancestor, okay? And that is the organism that we hypothetically call the last universal common ancestor, right? It's the thing to which everyone is related, all right? Now you can do reconstructions of what kind of genes that organism had, and it turns out to look like something very similar to a modern bacterium, right? So it's already a pretty complicated organism. It has the modern genetic code. It's able to copy DNA. It copies DNA into RNA, uses ribosomes to make proteins. It looks kind of like modern biochemistry, all right? So the point there is there could be a very long distance between LUCA and the actual origin of life, right? You could have something very different that itself evolved into something that ultimately had all of the common characteristics of contemporary organisms. Okay. So now we can think about scientists breaking this problem down into two approaches, right? One we'll call bottom-up, where you think about what is the permanent environment like and what kinds of chemistry takes place in that. And one we'll call top-down, where you look at what common features modern biology has and try to understand how they arose, right? And in principle, those two approaches ought to meet somewhere in the middle, which is the origin of life, right? Make sense? Okay, so here's the rub. Very simplified timeline of Earth history. Okay, so here you are here. This is today. This is the origin of the Earth about 4.55 billion years ago. We have certain mineralogical evidence that suggests the Earth had cooled enough for there to be liquid water on the surface by about 4.4 billion years. Okay, so pretty fast. Planet cooled, steam condensed. We had liquid water. We think liquid water is important for life. Going back in the fossil record, we can find generally agreed upon evidence for life from about 3.5 billion years ago, right? There's very few rocks that are old enough to preserve that kind of information. But what that leaves you with is a window for the origin of life that's, almost a billion years, maybe 800 million years, right? There's a very interesting paper that came out now that everything is digitized. They went and looked at how long every experiment that's been reported in the chemical professional literature was conducted for. All right, when you turned on the heating block and when you turned it off, right? That's the experiment. 98% was 48 hours or less, right? Because you set the thing up and at 5 o'clock you punch out and then you come in the morning and turn it off, right? Or you turn it on Friday and you come in on Monday, and we've kind of tailored our chemistry to be work over the time scales of our workday, right? So, if you're a graduate student, you don't want an experiment that takes 20 years. That's not good for you. So we don't really have a good conception of chemistry that takes place over very, very long time scales, right? We can extrapolate things like chemical kinetics, but there's that problem in there, right? That we're talking about a very long period of time. Okay, so that leaves us with the problem that we really don't know if it's a process that if you put the right things together on the right conditions, things come together and start copying themselves in hours or days or a week, or if it's really something that does take a billion years, that there has to be some slow accumulation of a particular compound or a particular ratio of some other compound that you'll just never observe in the laboratory because it takes too long. Okay. So I said it's very difficult to trace back what the early conditions on the Earth were like because the Earth is an active planet. We have plate tectonics that are constantly recycling the crust of the Earth. This is a map of the age of the oceanic crust. So if you don't know, this crust is created at these spreading centers along plate margins where there's hot magma coming up out of the Earth's mantle, cooling, and then being pushed along like a conveyor belt before it ultimately subducts at the next plate margin, right? So you have the very youngest crust where it's coming out of the mantle. The very oldest oceanic crust is only about 250 million years old, right? So the oceanic crust has been completely recycled, 16 times or something since the origin of the Earth. Likewise, there's a map of the ages of the continental crust. So this is the lighter crust that's managed to escape subduction. And we actually see that there are only a very few small rock outcrops that are older than 3.5 billion years old that have been preserved, because this crust is also sucked down over time into the mantle, right? There's a little outcrop in Western Australia, a little outcrop in northern Canada, and one in Greenland. And the one in Greenland is actually like the size of a VW bus. And enough, scientists have gone in with their hammers and chipped away at it that it's starting to be protected. Okay, so this is a point. This is a modern cell. Something like an E. coli highlights some features. There's a central coil of nucleic acid. There are ribosomes, various complex chemicals dissolved in solution inside the cell that are conducting metabolism. Often a multi-layered cell wall structure with a membrane, and then another wall, and then another wall, often motility appendages like flagella. They're pretty sophisticated little machines, right? Modern cell has a lot of moving parts. They're very precise. They fold very precisely. They are clearly the product of a lot of evolution, right? They've been tailored to work very well. Just an electron micrograph of the same thing to give you some appreciation of how much, machinery is packed inside one of these little balls, right? These are things that are a micron across, right? So a fraction of the width of a hair. There are, literally millions of ribosomes packed into these that are doing tremendous amounts of work to propagate the cell. All right. So let's think of, so theorists have been trying to come at this question, how can you think of something that's much simpler than a modern cell that might be able to do something like a living process, that it copies itself, it stores information about how you copy yourself, and it's able to do the work of copying, right? Because it is a machine. And there's a Hungarian theoretical biochemist who came up with the idea called a chematon, what he called a liquid machine. And the idea is that you would have chemical processes that basically do three things. You have a sub-process that interconverts building blocks and provides energy. You have a process that stores basically the blueprint of the system. And then you have a process that keeps all of those things together. So it's something like a boundary. And so you can recognize that as being an analog of a cell. It's an integrated system of subsystems that work together to recapitulate the whole. And now that you have this kind of template, rather than trying to build a modern cell, you can ask whether you can build chemical systems that mimic this kind of process, right? And they could do this using many different kinds of molecules, different kinds of processes. Okay, all right. So come back to this kind of question. Some things we do know about prebiotic chemistry. We have a bunch of sources that we can pretty reliably trust of simple organic compounds in primitive Earth. Are you able to start the first animation? Oh, I got it. Okay. Let's see if this works. So this is a view from the International Space Station. The orange lights are obviously cities and so on, but these little purple lights you see dancing around here are lightning storms, right? There's a lot of lightning in the Earth's atmosphere, right? I think you also see you'll see the auroras coming up. These are electromagnetic storms too, right? This is ionized atmosphere from the solar wind. There's a lot of energy coursing through Earth's atmosphere. When that energy passes through gases, it does chemistry, right? We know that lightning strikes actually generate a lot of nitric acid, for instance, from the nitrogen and oxygen that's in the atmosphere, and you can detect that in ponds after it's been a rainstorm. There are spikes in nitrite levels. Another source of organic compounds to the primitive Earth that we're fairly certain of now is the input of extraterrestrial organic matter. So this is a dash cam video from Chelyabinsk, Russia, from about 10 years ago, where an air-bursting meteor was caught on camera. Pretty big thing, also again about the size of a VW van, but coincidentally, which exploded over the city, and the shockwave basically shattered windows all over the city. A city like 200,000 people. No one was killed, but a lot of people suffered pretty bad glass cuts. So you may have heard of the Tunguska event. There's this kind of mysterious thing. The best likelihood now is that was also something like an air-bursting meteorite or meteor. Of course, there just weren't dash cams then to capture this thing. And it's actually a really interesting anecdote about this too. It turns out that slightly after the fall of the Soviet Union, when Russia started to do a little bit better economically, a really common scam was for pedestrians to throw themselves in front of cars and then say they'd been injured and sue people. And so there was this really rapid proliferation of dash cams to fight against these kind of scams. And we can thank that phenomenon for why we got this thing caught on camera. So the last really major source that people suspect may have been a source for organic compounds on the primitive Earth, still is today, are these spreading centers at the bottom of the ocean. So you have, they're along the plate margins at the bottom of the ocean. You have cold seawater getting sucked in above where hot magma is contacting the crust. That water gets heated by contact with that hot rock, and it undergoes all kinds of strange chemistry on its way back to the surface as superheated water. All right. Okay. So just to give you an example, we know all kinds of rocks from space that are just super rich in organic compounds and large varieties of organic compounds. The Murchison meteorite fell over Murchison, Australia in 1969. This was another serendipitous thing. It happened to be, I think it was a gold mining region that had been the mine had been shut down temporarily due to flooding. People were waiting for their unemployment checks outside the post office. Meteorite fell exactly at that moment. These people went out and just collected all this meteorite. There were about 70 kilograms of it, so it's a lot. This was found just about the time that the first samples were returned from the moon by the Apollo programs. And people thought the moon was going to have all kinds of organic compounds on it, and it did not have any, right? So it was a really disappointing blow to this theory of the early solar system being full of organic compounds. The Murchison meteorite kind of resurrected this whole idea, because here was a rock from the early solar system that just, you break it open, you could smell like diesel fuel, right? So this is a modern mass spectrometric analysis of this rock. It's estimated it may have something like between 100,000 and 100 million different types of organic compounds in it, right? So it's really just a zoo of different types of molecules. Going back to 1953, this was the year that Miller did his first experiment to try to show that the O'Parent-Haldane hypothesis really might hold water. And what Miller did was construct his now famous apparatus. It's kind of a microcosm. It's a sealed container. It has a chamber where you can put liquid water, which simulates the ocean. All right? That can be boiled, which generates steam. That simulates evaporation. There's a gas-containing portion of the flask. You can put any mixture of gases you want to simulate the early atmosphere. Miller used methane, ammonia, hydrogen, water. You have 2 tungsten electrodes, just metal rods that are melted into the glass. There's a gap of about 1 inch between them. And then there's a condenser here that returns the steam that's coursing through this atmosphere from the boiling back down into the aqueous phase, simulating rain, right, precipitation. And of course, you have an electric discharge generator, which creates a little spark that jumps across the gap here, like a spark plug, which simulates atmospheric lightning, right? And it's really not a humbug sort of experiment. Here's a simplified version of the apparatus. You can barely see the spark jumping there. This is right when you start the experiment. About 2 days later, you can see this stuff is just, there's organics dripping down the walls. The solution is changed to a brown color. There's been a large amount of atmospheric and aqueous phase chemistry of very reactive species generated by the lightning simulation that recombine and famously make things like amino acids, which happen to be major components of most biological organisms, right? And Miller was able to work out, as a chemist would want to, what the mechanism of this was. And it turned out to be a process that was discovered by Adolf Strecker slightly after Wohler's urea discovery, right? So this chemistry from the 19th century comes back and rears its head again. Okay, so Miller's demonstration there really kicked off a gold rush to see what other compounds you could make in these kinds of simulations. A scientist by the name of Juan Aro, working in Texas, showed that by simply putting hydrogen cyanide in water at a slightly basic pH, it would undergo a series of complexification reactions that ultimately gave rise to the biological purines, right? These are the nitrogenous bases in RNA and DNA, right? So pretty amazing. You just put a three atom molecule in water and shake it up, let it sit. It turns into this dark brown solution that's full of not only amino acids, but purines, pyrimidines, and a variety of other low molecular weight organic compounds. Just pay attention to this one, diaminomelionitrile. It'll come up again a little later. It's a tetramer of hydrogen cyanide. And so this has been a game that people have been playing now for since 1953, 75 years or so. Chemists have come up with more and more complex ways to try to understand how the molecules of life can be created, right? And they've had some success with some and not so much success with others. The ultimate goal of building a living system has been elusive. But there's a whole literature. It's considered a legitimate field of study at this point. But this is what we really just don't understand, this transition of how a flask full of molecules starts making copies of itself and gets self-organized and so on. One idea that's gotten very popular in the last 20 years or so is this idea of looking for autocatalytic reactions. So if you're not a chemist, a catalyst is a compound that speeds up a chemical reaction but doesn't get consumed in the reaction, right? An autocatalyst is the product of a reaction that feeds back and speeds up the reaction that makes itself, right? And so the difference there is that one catalyst becomes two catalysts, which becomes four catalysts, which becomes 8 catalysts, right? As you go around this reaction, right? It just speeds up in an exponential way, right? And you can think of different schemes for how autocatalysis would work depending on the stoichiometry of the reaction. So just showing you 2 different examples of that. There's a lot of ways a chemical reaction could be autocatalytic. And you can think of this, really simply, this is an analogy for biological reproduction, right? You mix A with B and you get 2B. If you mix, 2 rabbits with carrots, you get more rabbits. And as long as you give them more carrots, you get more rabbits, and so on and so forth, right? At some level, autocatalysis is an intrinsic property of biology, right? So this is a little simulation of what we'll call nest. It's a reaction diffusion model. So that means that molecules are being simulated floating around in solution. When they encounter each other, a reaction can take place. So it's governed by that diffusion process. And then we've got a second chemical process that is also autocatalytic that depends on the first autocatalytic process, right? So just to give you a simulation of what this kind of chemistry could look like. In reality, and there are starting to be chemical systems, actual chemical systems that can display this kind of behavior, right? So you're seeing the generation of these red molecules is dependent on the pre-generation of the green ones, right? From some environmentally supplied food stuff that is not visible in the simulation. And so this could have multiple levels down the way, where a replicator depends on another replicator to replicate. Well, I think it's neat. And so this is really, the frontier of this kind of study is trying to find chemical systems that can display these kinds of more complex replicative behaviors. A few years ago, we were looking at a system. Here's this dam again, where you have the oligomerization of cyanide being catalyzed by the addition of aldehydes, right? So this goes through a cycle where HCN gets fed in at different places in the cycle, and that kicks out this more oligomerized version of HCN. If you want to think of that, it's a cycle being fed by cyanide and producing diaminomelionitrile as a waste product. And the aldehyde is merely a catalyst. It gets reused in each cycle. Right? Interestingly, we found that this really simple catalytic cycle produces, in an autocatalytic fashion, little structures that self-assemble into little cell-sized balls, right? And that they seem to feed back. And as they are created, they create themselves faster, all right? So I try to give you, this is a movie of this process. This is a microscopic image that is about 2 millimeters in the X dimension. It's sped up, so this is about 5 hours of chemistry. And this is a simple reaction of ammonia, cyanide, and formaldehyde at room temperature. You know, molecules are so small you can't see them, so we're just looking at chemistry. But you'll start to notice these kind of little snowy things floating across your field of view. These are, in fact, molecules that are becoming large enough that they're no longer normally soluble in water. They start to aggregate. And you see this turns into a snowstorm of micron-sized objects that are catalyzing the formation of more of them. And you see this just gets going into quite a frenzy at some point. And at the end, you've got these large agglomerates of these kinds of things that, if you look at them under a scanning electron microscope, are actually spherical, micron-sized capsules. You'll see these kind of look like the eggs in Alien after they've released their payload. When you put these on a scanning electron microscope, it's a high vacuum, so any gas or liquid that's inside ruptures to the outside, right? And so these are kind of burst bubble structures. Okay, where does that leave us, right? That's a simulation. I know there might be many chemistries like that. We're interested in finding as many chemical phenomena as we can that do these kind of life-mimicking processes. Here we are, halfway out on the spiral, one of the spiral arms of the Milky Way. It's a really big question. If this process is really easy, there should be life like all over the galaxy, right? But that doesn't seem to be the case, right? We've been to Mars a few times. Venus doesn't look so good. To our best efforts to date, we seem to be alone, right? Unless you believe in UFOs. So we can talk about that. So famously, an American astronomer, Frank Drake, radio astronomer, came up with a way for estimating how common we might expect other civilizations to be in the galaxy that would also be able of communicating to us with radio signals, right? We call this now the Drake equation. There are modified versions of this, but basically what it does is it takes n would be the number of communicating civilizations. It takes into account the rate of star formation over time, the fraction of stars that have planets, the fraction of those planets that are Earth-like, that can have liquid water and so on, the fraction of those on which life actually starts, the fraction of those on which that life becomes intelligent, right? The fraction of those that life becomes technologically advanced enough to communicate through space, and then finally, the possibility that that civilization destroys itself. So what is the lifetime of that civilization? Or it's destroyed by its star or an accident or something, right? But for example, humans have been capable of radio communication for about 100 years, right? Give or take a decade or two. That's not a really long time, right? I personally believe we're not going to destroy ourselves, but there are those who do. so the important thing is when Drake first came up with this equation, you could really have wildly varying estimates because a lot of these factors were just wild-ass guesses, as we say, right? Importantly, when I started graduate school, which is 1996, there were no known planets outside of our solar system, right? And as of like last week, I think we crossed the 6,000 mark, right? So we know there are planets everywhere, and we look. We're starting to get a better idea of what the frequency of Earth-like planets is, and that's exciting. It's going to turn out to be a very large number. We really still have no idea if this origin of life process is easy or hard. It could be that it's just about impossible and we hit the lottery on this planet, right? Maybe there's a reason to believe that intelligence arises multiple times. We seem to see a few species that you could argue are intelligent on our planet. They're all pretty closely related, right? Vertebrates. We have no idea what the course of technological development is. We have nothing to compare that to. All right. So that's the paradox, right? Milky Way galaxy, it's about 100,000 light years in diameter. So this is the paradox of if there is life everywhere, why don't we see it here, right? Or why don't we detect it? And the idea is that if you could build spaceships, if a civilization could build spaceships that could travel at even 1/10 the speed of light, which maybe in the next couple of centuries, I don't know, our technology is moving fast, it would take about 1 million years to cross the Milky Way from end to end, right? And you can imagine, you build a sophisticated spacecraft, it goes to the next planet, builds a factory that builds 2 new spaceships and sends them out, and you repeat that process. In principle, if that were true, the galaxy should be full of aliens, right? Of technological aliens, right? Just for comparison, our furthest launched spacecraft, Voyager 1, is moving at about 1/18,000th the speed of light, right? So if using our technology that we've had since the 70s, it would take us, if my math is right, about 1.8 billion years to cross the galaxy, right? But the galaxy is 13 billion years old, right? So if something evolved 4 billion years ago, it's had more than time, right? So that is a question. I mean, in principle, if everything checks out, they really ought to be here, and they don't seem to be. So something in this equation is probably really poorly estimated, right? And I don't know which one of those things it is, but so yeah, just to give a shout out, this is absolutely amazing how quickly we're discovering new planets and new types of planets we didn't know existed before. I think, yeah, the current estimate is that about 10 billion habitable planets in the galaxy. Does that sound ballpark? Yeah. So this is where this leaves us, right? This is a take home. We really don't know how life started or evolved to get to the degree of complexity of LUCA. We know something about the timing, 800 million years. That seems about right. You know, in contrast to Miller-Urey experiment is like a couple a day experiment. So which is the hard step there. We know something about self-assembly chemistry. We're learning more all the time. Chemistry is a very vast and unexplored field. There may be a lot of planets where these processes can take place, but still we're on the only one we know of that hosts life, right? Lots of mysterious problems for the new generations of scientists to solve. And so with that, I want to just thank you all for listening. It's been a pleasure.
Thanks, Jim. So Vanessa and I have microphones. So if people have questions, we'll try to run the microphone to you so everyone can hear. Are there questions from the audience? One in the middle over there, Vanessa.
So on that slide you showed in the middle of estimates of Earth versus time versus year, there was this 2 billion estimate in the 18th century. Can you tell us anything about that?
If my memory serves, there may be a geologist in the room who would, I believe it came from counting sediment layers in a cliff, and where it was known how much sediment river floods deposited each year. And if you added them up, it came out to be like, it had to be something like 2 billion years. So,
Cool.
They were on the right track, right?
Right.
But,
All right. Thank you.
Yeah, you're welcome.
Well, first of all, thank you for your presentation. That was fantastic. The first thing that came to my mind was Kepler 22b. Given that it is mostly an ocean world, it's also rocky. How long would you say it would take life to form at, like, say, the very bottom of that ocean on that planet in the ways that life started on Earth? Like, do you think it's possible that life could be starting in that same way?
Yeah.
On that planet?
So there's kind of two big camps in the origin of life research field. And one thinks that these submarine kind of hot springs are the crucial environment for life to start. And the other seems to think that you really need something like a shoreline where water can evaporate and things can concentrate. Maybe light needs to catalyze something. If the shoreline people are right, then that planet like that is just not going to do you any good, right? If the bottom of the ocean people are right, then there's probably a lot of bodies in our solar system that are pretty decent candidates, you know, icy moons of the outer planets and so on. We just don't know, right? And again, it could be months, or it could be billions of years, and it's a coin toss whether even over that time span it does, right? So I know it's not a very satisfying answer, but you're welcome.
Thank you for the presentation. So I'm wondering, is there any evidence that we could form dipeptides, tripeptides, so like from the amino acids?
Yeah, that's a great question. So yes, the short answer is if you get amino acids concentrated enough, there's an equilibrium, and it's very far towards a single amino acid. It's maybe, I think, the equilibrium constant is like 0.01. So, right, 1% is in that state. And then that same equilibrium holds for the next amino acid. So there's 1/100th of that. So it's not a very efficient equilibrium. It's very far to the other side. The way life does it, though, is by pumping energy into the system, right? Using activated amino acids, and that draws the reaction over to polymerization. There are some chemistries that are known that can drive that. Not to the degree that biology does it, but yeah. Hi.
So on one of the slides, there were two scientists, and you said something about something falling into the water. What exactly was falling?
Okay. So go back a little. These guys.
Yes.
Yeah. So the idea was that, and this is related to this Miller experiment, that you had an atmosphere that was really different from the atmosphere we live in. 20% of our atmosphere is oxygen, and that's which we need to breathe. But that oxygen is produced by plants. So before you had plants, there wouldn't have been any oxygen, right? And that you can't really have methane and oxygen at the same time because they combust, right? That's cooking gas. All right, so it's possible in the absence of oxygen, you could have a high methane atmosphere. And then that methane could generate organic compounds through things like, atmospheric storms. And then that stuff would get dissolved in rain and washed out into the oceans. That was part of the model. Does that make sense? Yeah, good question.
I have two questions. One, in the Drake equation, what do we define as like an Earth-like planet? What are the sort of characteristics? And what kind of technology are we using, like right now, to detect life?
To detect life. Okay, that's two real good questions. So I think Earth-like planet is a kind of debated thing. For some people, it's a rocky planet that's about the size of the Earth, but I think people are willing to wiggle a little bit in diameter and rockiness and so forth. The other consideration would be that its distance from its star is appropriate to host liquid water, right, on its surface. Okay, so those are two things that have to be true, right? It has to be about the size and composition of the Earth and about the right distance from its star. Now, and bearing in mind that different stars put out more energy, right, depending on their size, so that distance can be different. I think on top of that, some people add the idea that it has to have an appropriately sized moon that stabilizes its tilt. Maybe that's like lower down on the categories. I think there are probably scientists who would add a few other criteria to that. Maybe like the presence of plate tectonics. That depends on a certain amount of radioactive decay in the interior, a certain mass and ability to trap heat and so on, composition. So I guess you can be more or less stringent. And then your second question was?
It was about what technology you're using
Okay. Yeah, so I mean, there's really two main ways. In our solar system, we're able to actually put physical spaceships in proximity or on the surface of planets and moons. We have chemical measurement devices that can actually see if there are molecules or certain types of structures present, right? With little remote laboratory devices. For these exoplanets, the hope is that you're going to be able to detect some gas in the atmosphere or some mixture of gases that really shouldn't exist. And you're going to be able to do that with a space-based telescope. So it'll be a spectral signal. Yeah. Because I mean these things are so far away, we'll never get there with a probe, right?
Yes, thank you, Professor Cleaves. This was fascinating. There's a lot of things that one might want to ask, but I was wondering if you could comment on, sort of philosophically, how important it would be if we were able to detect what we believe was definitive evidence for life, for example, on Mars. Not just that, ooh, there's life on another planet, but in relation to the Drake equation and the whole way of thinking about the possibility for life on other worlds, using our nearest neighbor as the test case.
So I think the discussion I've heard is that what you would hope is if you find life on Mars, that it's really different from Earth life, right? And the reason is that there's a pretty significant transport of rocks between the two planets. And unintuitively, when large enough meteors hit the Earth, they can actually bounce rocks into space, and conversely on Mars. And those, after wandering through space, will enter, right, the Earth's atmosphere. And we've recovered several meteorites from Mars, right? So some people think maybe life started on Mars and got transported to the Earth, right? It's kind of a minority view, but there's also the problem. We put tons of metal on Mars at this point. Have we possibly taken some bacteria to Mars? Like almost certainly, right? I don't think it's probably capable of proliferating there right now. But to your point, if we found a second instance of life on Mars and we were like, this is not terrestrial life, that would be huge, right? It's like two independent abiogenesis on two planets in the same solar system. I think that would throw the odds up quite a bit, right? So, yeah.
I guess kind of, sorry, like similar to that, I was going to ask about, there was like biosignatures found on Mars, like near like the Jezero crater or something. And I was wondering how we would go about studying those and seeing like, is this life different? Like, you know what I mean?
Yeah. So I know a lot of people who work on these Mars missions, and it's like one thing they always remind you is like it's really hard. You're driving this remote device that it takes there's a considerable lag time between when you say turn left and when it actually turns left, and they're really cautious about not driving this thing into a hole, right? So they do everything really slow. The instruments are they've been in space for years. They're doing their best. So they get back data at a kind of glacial pace, right? And stuff that if you could send a geologist with a rock hammer to Mars, you could get answers very quickly, right? But that's hard too. Yeah, so there was an idea that this rover in particular was going to cache samples around the surface of Mars, and then we were going to send a new mission back with a rocket that would collect them and then blast them back here where we could actually measure them. And I think it's still the rough plan, but I don't know that mission is like high priority because stuff costs money and yeah. So, but I think most people would agree we need to either bring something back or send something better there. So.
Hi, over here. Do you know the window of the temperature, pressure, or the physical extremities within which autocatalysis can happen? like what is the extreme temperature, or what is the coldest temperature, or extreme pressure, or least amount of pressure within which autocatalysis kind of reaction can take place, so that we can talk more about life on other planets, like let's say Jupiter or
Well, it's a good question. I think in your car's engine, when you combust gasoline, there are autocatalytic steps in that combustion, right? So that just phenomenon of autocatalysis isn't super duper rare in chemistry. But now our life as we know it on Earth does seem to have some box of parameters it can't live outside of. And is that going to be universally true for all living things, even if they're constructed in some completely different way? I don't think we know. Yeah.
All right, we're going to stop there because it's starting to get late. But I'm sure if you have questions that Professor Cleaves would be happy to stay for a few more minutes afterwards and answer those. So thank you all again for coming.
Thank you all.
And let's take our speaker.